thing. You cannot put a lot of slides at once. Otherwise, it's just too much for the thing to digest in one sitting. So, yeah, go by topics. Put at the most like 12 slides, and then ask the notebook LM to make that podcast. Yeah. Mm-hmm. Yeah. Yeah. The quiz is. Mm-hmm, mm-hmm. Yeah, yeah, I really, really like those features. And I was actually running out of imagination for questions, so I clicked on those quizzes for Notebook LM, and I really liked some of those questions. So some of them I felt comfortable enough to picking from there and putting it straight in the test. So so I'm giving you another clue on how to get those more practice questions. So, okay. You're welcome. All right, so here we're going to look at the further details on the complement system. So today is when we really start looking at those, strange floating proteins and what they're doing, and the interactions with other parts of the immune system. And this is an interesting thing about the immune system, like as a whole, because you have so many. Yeah. Did you upload this one? Yes, I just uploaded that. Yeah. So the thing is that we have so many moving parts that are interacting with each other, and sometimes you're trying to focus like in one aspect of the immune system, and then you see these other parts that have to do with something else, but they're also interacting here. So they're important for here, for there, for the other one. So at one point you really have to take a step back and see what is everything involved, and have I seen this factor, this protein before somewhere else. So if I did, it's like, what kind of function was it having before? What kind of function is it having now? How can it interact to really make the whole system a better defense, better systemic defense? Okay. So now we're going to see the details on how the complement proteins are working. And also, importantly, we're going to see where they are not working, and how is it that your own cells are defending themselves against possible attack by the complement system. Okay. Right? You don't see? I did put it up. Okay, let me pause it here. Just a minute. Okay, so we saw before that we have three branches for the complement pathway, or three pathways. And we have seen that they all converge at that C3 protein, right? Cleaving the C3 protein. So you have your classical pathway, and actually the classical pathway is called classical because it was the first one that was discovered, but it's actually the third pathway that comes into action, because this pathway depends on the production of specific antibodies, right? So this is the one pathway in complement that is also that depends on your adaptive immune response, right? So you have your classical pathway, and then you have the lectin pathway, and then you have your alternative pathway. And remember, the alternative is the one that is always surveilling for things. So today we're going to see how is it that the alternative pathway mechanistically is working, okay? All right, so here, we know that both the classical and lectin pathways, they use these adapter molecules that look like a spaceship or something with legs, right? So both of those pathways, what happens here is that you have these floating molecules, right? So this guy here is ficillin. Then you also have the mannose binding lectin. So they have those legs. They are ready to bind to patterns, right? That they can recognize. So we're talking about those pathogens, the PAMPs, right? So they're floating around and they are inactive. And then when they bind to those PAMPs, they change the conformation, because this thing is it's only like this part of my arm with the legs. Okay, so floating around. Usually they have 6 legs. So imagine I have an extra finger. Okay, so floating around. There we go. And when it's not active, it's just going like this. But when it binds to something, to the high density patterns, so when it binds to something, it's going to change conformation. So it's floating and floating and floating. Then if it binds only one, it's like, okay, nothing is doing, nothing is that. But when it finds the high frequency, it changes conformation. And when this conformation change, it sends a signal to the rest of that arm molecule that is going to activate the protease aspect of it. So that floating protein is going to start acting like an enzyme, okay? So when it acts like an enzyme, it is going to cleave some other zymogen floating proteins, and those zymogen floating inactive proteins, those are going to be C4 and C2, okay, or C2 and C4. When they cleave C2 and C4, okay, so this is both these pathways here, then you are going to get this thing that is called C3 convertase, okay? So C3 and C4, when you cut them, you are going to have, C2 and C4, okay? So when you cut them, you are going to have a piece of C4 that is going to fly away. This is going to be C4A, and a piece of C4 that is going to bind to the surface of the pathogen. Okay, so it's C4B and C4A. Okay, and then you're going to have C2, and that's the part that immunologists make sure that everybody gets confused. Okay. Because you have C2 that gets cleaved into C2B and C2A, but for some reason that I cannot figure out why, the people who are working on this and discovered this, they said that C2A gets bound to the surface of the pathogen and C2B flies away. Anyway, so when C4B that is bound gets together with the C2 part that got bound to, then you get a C3 convertase. So some textbooks say C3 convertase of these 2 pathways. They say that this convertase is called C4B2A. Okay. More recent textbooks say that the convertase is called C4B2B. Okay, and of course I'll never ask you what is the C3 convertase on a test, bless you. I'm going to say, and I will not put the answer saying that C4B2A, C4B2B, that would be not fair, okay? So what do you have to know? Here, just by convention, we're going to call it C4B2A, okay? So if I ask you what is the C3 convertase of the lectin pathway, you should say C4B2A. What is the C3 convertase of the classical pathway is also C4B2A. Okay, so these 2 pathways, they use the same C3 convertase. Okay. Now, when you look at your alternative pathway, that guy uses a different C3 convertase. He uses a C3 convertase that is called C3BBB. So that one is a bit easier to remember because you just get stuck on the B's. Okay, C3BBB, that's the convertase, C3 convertase. Okay. So C3, cutting C3 is the conversion of those 3 pathways, like also as we have seen before. And here we go for the first question. Okay, so here the alternative pathway. Okay, so let's see what is the answer here. this one we already had before, so this idea of your alternative, yeah, you have a question? No, okay. So the idea of this alternative pathway was already, you know, part of your first exam, right? But here you get extra points for it. Okay. Do we need more time? No? No? Okay. All right. Is your answer B? Yeah. Come on. Oh, I have to stop it. Okay, here we go. Okay. All right, so C3 cleaves itself, and that's the pathway that is always active, and now we're going to see how that happens. Okay. So the main idea that you have these proteins that are inactive, and they are, you know, floating around in the Zen form. That's your meditating Zen guy floating around in your blood, not doing anything. Okay. So they are inactive. They are these zymogens, okay? If they get activated for no reason, they could cause damage, right? Because once you let loose the cascade path of the complement, you can end up with holes on the cell membrane, right? You can end up with pores that could destroy cells that are part of your own cells, and you don't want to do that, right? So they could destroy the host, all right? So you have several defense mechanisms that are going to prevent damage to your own cells, okay? So the question here, when the cascade is activated, like we have a spontaneous activation in alternative pathway, so when it's activated, how is it, or why is it that you do not destroy host cells, okay? So what happens here? When you activate the C3 just by itself, right? So C3, active C3 can get inhibited by some molecules that are working on your cell surfaces, okay? So inhibition is one of the mechanisms, okay? Another mechanism is the very short time that the active protein floats around, okay? So having very short active period, it ensures that this protein is not going to be hanging around for too long to cause damage, okay? Besides that, it will also ensure that the protein is not going to have enough time to fly from one side to another side on your body to cause damage, right? So short half-life is really giving you a time limit and a space limit, okay? So it has temporal and spatial type of control of possible damage, okay? So is that clear? Okay. How that thing is not really damaging right away, okay? So now let's see more details how that is happening, okay? So here are some molecules that are very important to prevent formation of MAC on your own cells, all right? So you have these control proteins, okay? Control proteins that are expressed, and some of them are on the surface of your cells, and some of them are floating around, okay? So basically, those control proteins are going to avoid the damage or minimize the damage, okay? So here is an example of a control protein that is called decay accelerating factor. Now, if you think about the name of this protein, decay accelerating, it means that it's going to speed up the breakdown of actin complement proteins, okay? So what is going to happen here, this decay accelerating factor, when it finds C3b and also C4b that are stuck to your own healthy cells, this thing is going to block the action of C3b and C4b, okay? So now you're going to block the activity of those convertases. So you're going to stop the cascade, okay? So here is the idea of this decay accelerating factor that is, abbreviated as DAF, And one thing I have here to remind you that you have a protection factor is this guy here. Okay, so you got here Daffy Duck defending you. So that's Super Daffy Duck defending you on the surface of your own cells. All right. So let's see what is happening here. Okay. You have at first you have possible binding of C3b. Okay. That could come with another factor that is BB to form a C3 convertase. This can mean damage to your cells. Then this is from your alternative pathway, and then you have this other C3 convertase that comes from the other pathways, right? If it is left on your cell, that's a big problem. You're gonna get many holes on the cells, right? So what happens here, because you have DAF already expressed on your cells, DAF gets there and interacts with the bound portion of your complement proteins. So DAF gets there and blocks the formation of the C3 convertase on the membrane. Okay, so this is going to stop the cascade, like right in the beginning. Okay, is that clear how that defense protein works? If you have that on the surface of your cells, you're not going to be forming the C3 convertase to amplify the cascade. All right. So here is another factor that is important. Okay. So it's also a part of your complement control proteins. Okay. So these guys here, you have this factor floating around. This is a protective factor. Okay. So you have this factor I floating around, and one way that you remember that this thing is a protective factor is that the I is for inhibition. Okay, so factor I is going to inhibit the cascade somewhere. Okay, so what's going to happen here? You have factor I floating around, but by itself is not going to do anything. It needs to associate with a cofactor. Okay, so factor I flies around, and then it needs to find a cofactor. So it can associate with this cofactor that is called membrane cofactor protein. Okay, so cofactor protein on the surface of the cell, just like Daffy was. Okay, or you could have factor H that is floating around and not showing here, or you could have a receptor on the surface of the cell that is called CR1. Okay, CR1. Yeah, we have seen CR1 somewhere else, but here is also important for this defense mechanism. Okay, so what happens? Factor I associates with one of those cofactors. When it is associated, it is able to interact with C3b. Okay, factor I here associated with a cofactor on the membrane. Now it interacts with C3b because this has to be your defense mechanism. So what it's going to do is going to inactivate C3b. Okay, that's with the I for the factor. Okay, it's going to inactivate C3b by cutting a piece out of the C3b. Now this C3b is not going to accelerate the cascade. Okay, you are not going to have promotion of the complement cascade on your own cells. Okay, what is the use for this thing here? Okay, because after you cut a piece out of it, and actually this is not even the whole story, you also have another piece that's called C3d, but let's not think about that now. Okay, so after you cut a piece of your C3b out, another piece stays there. Okay, so this is inactive for formation of the MAC, but it has certain functions. All right, so what happens here with your C3b, it works in different ways. Like, for example, it stays on the surface as a tag. So this thing, even if it's not going to continue with the cascade, it is working as an opsonin. Okay, so it's tagging the surface that it bound to. Okay, so what happens here is that it helps to remove tagged microbes, even if the microbes also have other parts that are with active cascade complement. Okay, it is going to help also to clear up antigens and antibodies complex. So those are called immune complexes. Okay. Besides that, they can be attached to cells that are dying. They're undergoing apoptosis. Okay. So presence of this tag is also going to help you clear up the debris. Okay. And besides all that, you also have a part of the immune system that is going to teach damage control, meaning tolerance branch of your immune system. Okay, so there are several types of functions that this inactive part of C3b is important for. Okay, so what's the main idea in this slide here? You have this floating factor that is a protective factor that interacts with a cofactor on your cell membrane, and once they interact, they're going to inactivate C3b. That's the important part of this. And I was looking for a superhero for that one, and I couldn't find anything. I put MCP on Google, and some superhero showed up. I have no idea who that guy is. But if it helps you, if you identify this thing, you know, it's help us out. If you have other ways to remember this thing, fine with me. Sure. All right. So here, this is just, I have several kind of repetitive or redundant slides, because sometimes the whole notion, the whole idea of how this thing is working, it kind of takes a while for us to digest. So I mentioned it several times. Maybe one of those slides is going to click something in somebody's memory or brain. Okay. All right. So here, proteins are protecting the host. Okay. You know that you're going to have here your factor I. Okay. And remember, I mentioned the other cofactors, right? So besides the other cofactors, you also have floating around this other guy that is factor H, right? So these guys here will trap C3b even before it binds to the cell surface and inactivate C3b, like right there, right before it interacts with anything, okay? So what is important here to know? Okay. So we know that you have the C3 convertases, right? The C3b BB. We're going to see exactly how that works. Okay. Let me see what makes sense here. Okay. All right. So this, let's go forward with the rest of the explanations, and then you're going to go back and associate with the mechanism. Because this is a tricky part of all this complement system. There are so many moving parts that sometimes it's difficult to put everything in a reasonable order, right? So here are some of the moving parts. And you have about 30 or so of these moving parts, but we're only concentrating 10 or 12 of them. Okay, so 9 of them are complement something proteins, okay? C something proteins. So here you have them. You have your C1. Okay. C1 looks like a strange cucumber with legs, right? And they have 2, 3, 4, 5, 6, 7, 8, 9. At least they are in successive numbers, right? But you got to know the personality of these components and their functions, okay? When they get involved and really what are they doing, okay? So as we're going to see, your C1 is very important for activation of your classical pathway, okay? So C1 is going to interact with antibodies, all right? Then you have your other protein here, C2, okay? C2 is important for making the C3 convertase in the other two pathways, okay? Then you're going to have here C3A, C4A, and C5A, and those are the pieces that fly away, right? 3, 4, 5. The pieces that fly away and not bind to the surface, okay? So when those pieces are flying away, they are not really flying too far. They are flying away close by because they have to do some very important activation processes, like anaphylotoxins. Okay, so these proteins are very good at activating degranulation of some of your innate cells. Okay. Besides that, they work as chemotaxins. They are going to attract leukocytes in general to get to the area. Okay, so they're going to attract white blood cells to get there. So not only they are attracting the cells, then they are also activating those cells to do their jobs once the cells get there. Okay, so C3A, 4A, and 5A are very important for those functions. All right. So again, as I mentioned here, your C3b, that's a very, very important guy because that's the guy that tags. So opsonization is a function of C3b. And you also have C3b that is going to act later on for formation of your MAC. Okay, so C3b is going to be important for cleavage and activation of C5. It's going to be like a C5 convertase. Okay, all right, so that's a bit of a C3b. Then you have here, okay, C4b, okay, C5b. Okay, C5b, once it gets cleaved, remember with zymogen, it's going to get cleaved. C5b, you have one part of C5 that flies away, that's the C5a. Remember, C5a has those functions. And then the part that binds to the cell surface, that is a very important part. It's going to put the membrane attack complex together. Okay, so C5b is going to attract the other guys, 6, 7, 8, and 9, to form the pore on the surface of the cell. Okay, so here you have 6, 7, and 8. They are part of the pore of the cell. They are able to form a pore on their own, okay? But when they get together with C9, then you really get a potent pore on the surface. You get a lot of C9s, they get there and punch holes, basically. They go 9, 9, 9, 9, 9, 9, 9, and punch a lot of holes to make, you know, the surface, to make a big hole on that surface, okay? But one interesting thing about this is that if you have a person that, for example, is not expressing C9 in their genome, this person still has active complement system and MAC formation. So C9 is, you know, is cool to make a neat pore, but is not really vital for the function of this system. Okay, so these guys here can form a pore, but then with C9 they make it better. Okay, and notice that from C6 to C9, you don't have cleavage of those guys anymore. You just have recruitment of those guys. All right, so you're going to see cleavage of the zymogen forms from here to there. These 4 guys over here, they get activated by cleavage. The other guys, they get activated by recruitment. Okay, they just brought there together. Once they're brought together, that's it. They do their jobs. Okay. Okay. So besides all that, you have also, okay, these guys I'll mention in a minute. You have also this protein that floats around here. That's called properdin. Okay, so this guy, properdin, is another form of defense because it is not going to interact with the complement system that binds accidentally to your healthy cells, right? But what it does is that it interacts with C3 and with C3 convertase that bound to the surface of pathogens, all right? So when you have your C3 convertase that bound to a pathogen, then properdin gets there and stabilizes that interaction to really make sure that your C3 convertase is going to kill the pathogen. Okay, so this is a type of enhancer of function of that C3 convertase. Okay. All right, so that was like a overall idea of all these complement cells, right? So now we're going to look at some mechanisms of how that thing is working. Okay, so first thing we're going to see is your alternative pathway. We know C3 is always floating around in the zymogen form, but then it can activate itself spontaneously. So what is happening here? Okay, you have in the inside of this protein, you have this hydrophobic core, right? Everybody took cell bio? Yeah. So remember, like protein folding and hydrophobic core and hydrophilic exterior of the protein. You know what I'm talking about? Yeah. Okay, so inside this protein, you have this hydrophobic core, and usually, it's not really exposed to water, but once in a while here, you're going to have a bit of a conformational change, and when that happens, you're going to expose a part here that has a thioester bond inside of that core, and that thing can be attacked by water. Okay, so if water gets in there and attacks that thioester core, you're going to end up with C3b and C3a. You're going to cleave the protein. So just by having water in this place, you're going to get a baseline autocatalytic action of C3. Okay, and is this thing like floating in water to start with? Do you think you're going to have water in the environment where you're going to find C3? Yeah, where do you find C3? Where? Extracellular fluids. Do you find it in the blood? That's the wake up question. Yes. Okay. Yes, that's the surveillance protein in the blood, right? And do you have water in the blood? Yes, it's water-based, right? So yeah, you're always going to have this baseline autocatalytic cleavage of C3, okay? So then what's going to happen? You can have C3b that can be neutralized just by extra water that is floating around, or if C3b is very close to a cell surface, it can bind to any cell surface, right? But this is when your defense mechanism has to be at the cell surface, because you don't want to get holes because C3 bound to it, C3b bound to it, okay? So if it happens that it binds to your own cell surface, then you get you inactivated, okay? If it happens that just by chance this thing bound to some bacteria that was not supposed to be there in the first place, then that's a good thing, because those proteins, C3 job is a surveillance job, and it doesn't know if it has bad guys or not. It's just, getting activated just because it can, and if it binds to something that the immune system didn't see, so then this thing is going to kill the pathogen, okay, by activating the MAC. Besides, you're also going to do opsonization of the pathogen. Okay, so if it happens that pathogen is around that was really sneaking away from immune cells, right, just floating in your blood, then this guy can catch it, can tag it, and can destroy it. Okay. All right. So remember here on the surface of the pathogen, this interaction is going to be stabilized by the other floating protein that is your properdin. Okay, properdin right there. All right. So here, in more details, what is happening here with your C3. Here is when you have this initial cleavage, right, autocleavage, and you have the two pieces that are getting separated. And here is when you have this thioester bond that can undergo a nucleophilic attack and covalently bind to something else. Okay. If it is water that attacks this, so C3b is going to be inactivated, it's going to be just floating around. Okay. If it happens to be something else, like for example an amino group or a hydroxyl group that is part of a protein on the surface, or a part of some carbohydrate on the surface of a pathogen, right? So this reaction of a nucleophilic attack is what is going to covalently bind this C3b to that surface. Okay, so once it's bound, it's really, really bound. You're not gonna take it out of the pathogen surface. You're just gonna tag that pathogen for action by something else that's gonna get there and eat it. Okay. All right, so let's see what else is happening here, right? So that was the story with the C3b getting on the surface, okay? So now let's see what is happening with the rest of the C3 that is getting the MAC activated for the alternative pathway, okay? So C3 is floating around, and let's say here C3 interacted with water, okay? After C3 interacting with water, you have this, okay, you call IC3 form, okay, but anyway, it's interacting with water. Basically, it changes the conformation a little bit in order to allow interaction of C3 to this other floating factor, okay? This floating factor is called factor B, okay? So if you don't have initial hydrolysis of your C3, then it's not going to interact with factor B. But when it interacts with water, now you allow it to interact with factor B. So factor B interacts with that. This interaction is going to change a little bit the conformation of that B protein. You see the little bit of opening in the site over here of the B? That means the B that is bound to C3 is now going to act like a attracting enzyme, okay? So B here is going to attract this guy, the enzyme, that is called factor D, okay? Factor D is going to go right there at this cleavage site and is going to cut a piece of your factor B, okay? So D here is the enzyme, cuts a piece of factor B. Factor B is part of your complement system. So you're going to have two pieces that are going to come out of it. One piece is going to stay bound. The other piece is going to fly away. Okay? With the piece that is stay bound, here you have your C3, IC3 bound to BB. Okay? Now what's going to happen here? Your IC3B bound to BB, remember you have two options for this complex here. One of them is to stay flying around. Another one is to cleave the A part of your C3 into C3A, and the rest of this thing can be bound to your cell surface. Okay, so here you can have, this is missing actually a piece. You can have, here is the other part, okay? You can have your C3B, BB that is bound to the surface, okay? Besides that, you can also have this guy here as an active C3 convertase. So you have it active floating, you have active bound to the surface. Okay, so what is the result of this? Once you have this complex that is formed of C3b, BB, Remember, this guy here is your active C3 convertase. It is going to amplify the cascade at this point, okay? So then here is a better picture of this. So let's see what happens here. From a different angle, if you have C3b bound, you can also interact with the B floating around. D goes there, cuts a piece of the B, and here you have your C3BBB convertase. This guy is telling you it's going to convert C3 into bound and floating pieces, and this is your amplification part of the cascade. So this one guy here is going to convert thousands of C3s into C3A and C3B. Okay? So one enzyme here is going to recruit a lot of C3s to get a lot of C3Bs and a lot of C3As right at the site of your pathogen. Okay? This process of amplification is really a process of a positive feedback amplification. Okay? So formation of this is just going to recruit more and more and more of the C3 that is really going to tag your pathogen. Okay? Is that part clear so far? Yeah, question. Okay. Yeah, I know it sounds like an alphabet soup. So let's let's get a picture here. Let's see here. Okay, so this here is factor B floating around. Factor B. Okay. Now this here is C3. And what's going to happen? C3 can autocleave, floating around. It can autocleave or can interact with water. Let's say here this guy autocleave, okay? So a piece of it is C3A is going to fly away. Flow away. C3A flying away, C3B, and here is a pathogen. C3B goes in here and goes like, oh, I'm bound. This guy here, what is this guy again? B, big B, okay? So C3B got bound over there, that's it. Big B comes around and like, oh, interesting guy, let me interact with it. But nothing's gonna happen because this guy is inactive. But now it got a bit of a change in the conformation. There's like, somebody could come here and cut the piece off. So there comes this other guy. This other guy here is factor D. Okay, factor D goes there. It's like, I could go there and cut this thing off. So it goes here. That's it. Now this guy here is active. That guy over there is C3 convertase. Okay, C3 convertase is there and is active. Now this guy here is an enzyme. If you have more of C3 floating around, when it gets here, the C3 is gonna be cut by the convertase. Okay, so this is the part that you get C3 coming around and getting cut and getting stuck on the membrane. Okay, so yeah, I know it's, the alphabet soup of this thing is annoying, but just think about the molecules and what they're doing, how they are floating around, and where they have to go in order to be active. Yes? Yeah. This guy here is the same as this guy here. The only difference here is that it is acting on C3. C3 got together here, and this guy is going to get cut. So this here is going to go here. Okay, so let's put a tag. So, okay. So here, let's put a dot on this guy here to trace it. This guy here got cut, and a part of it went on the membrane. And then if you put another trace on this part over here, part of it flew away. Okay? and then you're going to get another C3 that gets there, and it's going to get cut, and a part is going to get here, another part is going to fly away. So you see, like here, you're going to have 4 molecules of C3 that are getting 1, 2, 3, 4, and those are going, so let's try 2, 3, 4. So those are going here, 2, 3, 4, and there's 2, 3, 4 pieces. One at a time, it gets there and gets chopped. Okay, so this is the part that you have the amplification of this thing, like cut and cut and cut. So this guy here is your C3 convertase. Yeah.
So basically, this is the way to do C3B from C3
Yeah, yeah. Right. Yeah, it's just amplifying the presence of your C3b over there. Okay, and C3a flying around. Okay. So remember that in order for this to work really well on the surface of your pathogen, you need this guy that is going to stabilize your C3 convertase. It's going to make it really useful and live longer on the surface of the pathogen. So this thing is not going to interact with your C3 convertase that is on your own cell surface, because that C3 convertase on your own cell surface is going to interact with something else that's going to inhibit it before this guy has a chance to get in there and stabilize it. Okay? So remember, you have your Daffy Duck, you have your MC3 superhero. Those guys are already present on your own cells. So those are going to stop it before this thing can be stabilized. So this is really important for stabilization on the surface of pathogens. All right. Okay, so we have that. We have your C3 convertase. For this guy, when he gets activated, yeah, it is waiting for them. Okay, it's just before it was not active. It didn't care who was around. When it get activated, it is just waiting for the substrate to get there. Okay, so C4 and C2, they are already floating around because they are part of whatever is floating around in the blood. Okay, but because now this enzyme is active, when C4 gets there, it's going to get cut. So this thing is active. C4 goes around and says, "Oh, I got cut." C2 is floating around and goes, "Oh, I got cut too." Okay? So just because the enzyme gets activated, it can now start cutting what the thing was already floating around.
C2B will have a higher chance of binding instead of waiting for the pathway to actually start breaking and start attracting the binding.
Okay, so C2B. C2 is the weird guy. The C2B part flies away. C2A binds. Okay? I don't remember what is the function for the C2B that flies away, because all of these pieces have some functions, but it's just too much to remember. No. What you have to think about here is that once you get C4 and C2 cut, the parts that bind to the membrane, they are going to form your C3 convertase. And that is the amplification step for this pathway. Okay? So amplification step, you get a lot of your C3 bound, C3b on the surface, and then it goes down to the MAC formation. Okay? Okay, so here is just showing you like this little picture of your mannose binding lectin. And here is showing you the part of your serine proteases that once you bind, you're going to have conformational change and activation. And then you're going to have here your C4 and C2 that is going to be cut and then interact with the surface of that bacterium. Okay. So here, we look here at this guy, mannose binding lectin with the arms, right? And here you have your serine proteases, right? They're going to cut C4 and C2. the same idea, you also have this other guy here that is a cousin of your mannose binding lectin. So this other guy here is ficolin. Okay, so ficolin and mannose binding lectin, those are cousins. Okay, so what happens here? Mannose binding lectin likes to bind mannose. Okay, and then you look here at ficolin. Ficolin likes to bind to an N-acetylglucosamine, which is part of bacterial cell wall. Okay, so these two things here are part of the same pathway of mannose of binding lectin pathway, lectin type of pathways. Okay, now comparing these two structures here with this other guy over here, this guy over here is called C1. More specifically, is a C1q. Okay, so this guy here, it looks like that, but it doesn't have the MASPs. What this guy has, there are other C1 proteins that are interacting with that. So these small round C1 proteins, they are also serine proteases. Okay, when this thing is flying around, is not interacting with anything, so your serine proteases are quiet. When this thing interacts, not directly to the surface of the pathogen, but when it interacts with the constant region of antibodies that recognized the pathogen, when it binds, now you're going to have conformational change and activation of those serine proteases. All right, so this complex of C1 is actually C1QRS. Okay, so C1QRS, the RS part, those are your serine proteases. Okay, so here let's see how this thing compares. Okay, same. The mechanism is about the same as with your lectin pathway. All right, so you have this spaceship thing, activate serine protease. You're gonna cut C4. You're gonna cut C2. And you're going to get your C3 convertase. Then C3 is going to go there, and you know the rest. Okay. Compare that with your classical pathway. First thing that you're going to do, you're going to have to let the legs interact with antibodies. Okay? So here, at least 2 of the legs need to interact with antibodies that are bound to epitopes that are close to each other. So same thing, when you have the conformational change, this is when you're going to get activation of your serine protease. Okay. Now, important thing to know, there are only 2 classes of antibodies that are very important for activation of this pathway. Okay. They are IgG and IgM. Okay. IgG, as you're going to see, probably not today, but as you're going to see, IgG is a monomer. So one molecule floating around like this. Now IgM is a pentamer. So you have 5 antibodies that are held together and they're floating around like this. Okay. Mechanism of activation of these antibodies is the following. When you have here your IgM antibody that is a pentamer, okay, 5 antibody molecules connected. When IgM recognizes the surface of your pathogen, it goes from flat to kinked, to changing conformation of the antibody, like that. Okay. When it goes like that, then it exposes the constant region out of the flat plane. So now the constant region is kind of like bending upwards, and that's when you have the leg of your C1q that is capable of interacting with that constant region. So now, if you have here your C1q that is floating around, it's going to interact and it's going to change conformation. Okay? So this is a mechanism of activation of your C1q. Okay? So this was your IgM bound, and this is your C1q bind. Now you get activation of C1q. comparison here with IgG tagged pathogen, okay, you're going to have the same idea. So now you're going to have two antibodies that got stuck to the surface, and here your CO1Q. If it binds only one, it's not going to get activated. But if it binds two, that is your conformational change. Now the thing gets activated. Okay. So what is the serine protease going to do? So here's the idea. Okay. Here's your CO1Q. Binds to at least two constant regions on antibodies, okay? And you are going to have activation of the serine proteases, and look at the complement proteins that it's going to cut. It's going to be C2 and it's going to be C4, and you have seen that before, right? So same thing, you're going to have C4b that binds, C2a that binds, and this guy is your C3 convertase. And you're going to get amplification of that cutting of C3, okay? So one molecule there is going to cut a lot of C3s. One molecule here is going to cut a lot of C3s. And this thing is going to make a lot of C3 convertases, and this thing is going to make even more C3bs to tag the pathogen. A big, big amplification. Everybody was cool with that? Okay, now let's see here what else is happening, okay? So once your pathogen is tagged with C3b, okay, this process is called opsonization, okay? And this is very important for pathogens that, especially bacteria, that have a kind of a gelatinous coat on top of it, okay? Because some bacteria, including gram-negative bacteria, they have an extra membrane to them, right? They like to hide from phagocytes. So if they are hiding in getting away with things, they can really make an infection go really bad. But if they are tagged by C3b, they can't really hide that much, okay? So tagging of the C3b is going to attract phagocytes to interact with the C3b and eat up the bacterium, okay? So this is a very, very important opsonization aspect of the system, okay? So now let's see what happens here to the rest of the cascade, okay? So you have here your C4b2a, that's your C3 convertase, right? You cut C3, you get C3b right there, right? When you put these three guys together, C4b, C2a, C3b, you get another convertase. You get this guy that is called C5 convertase. Okay? Now what's going to happen now? C5 is going to show up, and you're going to cut a piece away from C5, and you're going to get C5a and C5b. C5b is going to get stuck on a membrane. C5a is going to fly away. Okay? So the name of the convertase here is C4b2a3b. Yay. 3 parts to the convertase. Okay? All right, so let me see here. I mentioned that before. Oh, okay, so let's get another point before we go further to the MAC thing. All right. Question here. What is the first event that takes place on the activation of your classical pathway? Okay, so the options here are C4b2a is bound to the surface. No, that's already your C3 convertase, so no. Okay, C1r is activated. Oh, this guy is a serine protease. That does not happen first. C3b binds to C3 convertase. That comes after activation of C3 convertase. C1 complex, the flying cucumber with legs, bind to a constant region of an antibody. That sounds interesting. C1s cleaves those guys. So I just gave you the answer. Does anybody need more time? Two people are still sleeping. I mean, if you're in class and right after the test, I want you to get all the points. Change your minds quickly. Okay, what's your answer? D. Okay. All right, so here is that idea of your classical pathway, right? So you have your cucumber, binds to antibodies, activates certain proteases. You're going to get C4A, C4B, C2A, C2B. You get your C3 convertase, and then you're going to have your, how did C5 get there again? Oh, C3B. C3B got together, made your C5 convertase, and now you're going to get C5. C5 is going to go to the membrane, and now the attack complex happens. Okay, so let's see what happens here. C5b gets there, okay, binds there. So this is bound to the complex here. Gets there, and it's going to attract its friends that don't need to get cut. So these guys here, C5b, once it's in the complex, is going to get 6, 7, and 8. 6, 7, and 8, they're going to get there, interact with C5 that is bound, and they're going to make a hole on the membrane. The hole is enough to kill, but just in case it didn't kill enough, it recruits this big guy here that is C9. Okay, so C9 is going to get in there and insert itself on the membrane and make this big tunnel, okay, big hole on the membrane. Look at this guy here. There is a protein on the surface of your cells that is called protectin. Protectin to protect you, okay? Protectin is not properdin. I know, it sounds same, but not. Properdin helps you kill pathogen. Protectin helps to protect you. So what protectin is going to do, it can block this C9 pore formation. Okay, so this is another safety mechanism for your cells. Okay, another name for protectin is CD59. Okay, same thing. All right. So now let's see here what is happening with your MAC complex. So here we go. You have your C5b that is bound. You call on the friends 6, 7, 8. So 6, 7, 8 is enough to make whole, but then they call on C9. And then C9 really gets together. There are like a lot of C9s, okay? 9, like 1, 2, 3, 4, 5, 6, a lot of them. Go there and really make holes on the surface of your cell. Look at the structure of this thing. A lot of C9s, they get together. So C6, 7, 8, 9, those don't get cut. They just float around and they interact. When they interact, they're going to form the pore, okay? But they only brought into the picture after you had already cut C5. So C5b, yeah.
Question, not related to this, but related to the previous slide. About the pathogens that are already going around, and then C3b is being attacked. It comes to mind what would happen when biofilms comes in.
Oh, biofilms are a totally different beast because they make their own enclosure complex and they are shielded away from complex attack, from complement system. Okay, so biofilms are quiet places for bacteria. All different types of bacteria get together in biofilms, and they are dormant. So they are not really susceptible to some drugs like antibiotics, because antibiotics work when the bacteria is dividing, or at least when they can reach the bacteria. And those in biofilms, they are really being kept away from the rest of the fluid environment. Okay, so biofilms are a big problem. Okay, so only when the bacteria gets out of that cocoon of biofilm, then the bacteria gets susceptible to attack by complement system. Yeah. All right, so everybody got it so far here, formation of the complex. All right. So how is it that this complement system is working? You know that this is surveilling all the time, so it's part of your innate system. You know that interacts with antibodies, so it is part of this interface between innate and adaptive system, okay? And also is going to help with the overall final functions of the two systems, innate and adaptive systems. So let me see what I have here. Okay, so main functions of pieces of the complement system, okay, that you should actually be familiar with, okay? The components and their functions. All right, so you have here C3A. Okay, what is C3A going to do? C3A is going to help cells to degranulate. Which cells? Well, the ones that have granules. So you have inflammatory cells like mast cells and also basophils. Those are inflammatory cells. When they bind to the flying C3A, they are going to dump the granule content. So C3A is important for degranulation. Okay? C3A is also a chemotactic factor. So it's going to attract the cells to get there. Okay? This property is called, what is the name of the property? No. I forgot. I'll remember in a second. Okay. So it has chemoattractant property, and it has also this other property that allows degranulation. Okay. All right. So here is the other part of C3. C3b, important for opsonization. All right. Tagging and interacting with other receptors. C5a is also important for degranulation and chemotaxis. All right, so this guy is also chemotaxis, but C5a is the more potent chemotactic protein. Okay. All right, so here we have a membrane attack complex. Oh, here we go. You have all these proteins here. C5a and C3a, those are important for degranulation. C5a is most potent for chemotaxis, but C3a also. Okay. C3b, we focus here on C3b, okay, is important for opsonization. Then you have your membrane attack complex. C3b is also important because it has receptors. This is very unusual, okay? It has receptors on the surface of red blood cells. So C3b can interact with receptors that are right there on the surface of red blood cells. So this gives a very important function for red blood cells as agents of your innate immune system. Okay, yes, I know, I'm blowing the time. Okay, so we're going to stop in this one, but keep in mind, your red blood cells can also clean up tagged pathogens from your blood. Okay? Tagged with C3b or opsonized pathogens. Okay? And this is a function of red blood cells that we usually don't think about. Okay? Clearance of opsonized pathogens.